Before I hand it over to Dr. Xu, I would like to make a quick overview of the PacBio's population genomics offering in the context of a precision health initiative. So PacBio really actually made a huge progress in terms of overhauling our system to enable high throughput sequencing for the human genome. And last year, towards the end of the year, we actually launched the system, which is called the Revio. And this system basically inherited all the benefits we actually had on the prior generation of the systems in terms of the read length, the accuracy, the uniformity of the sequencing coverage, sequencing the native molecule in order to actually ascertain the epigenetic information, which is very important in all aspects of genomic study. And the hallmarks of this system is basically these four important features. First, we are actually enabling for the first time to actually sequencing a large number of genomes in a given time period. So, for example, right now the system can actually allow you to sequencing up to 1,300 human HiFi genomes per year, and we made significant progress in terms of streamlining the workflow. Makes it extremely easy to use. The computational powers, also enabled by the onboard computing, so that you can actually get very, very good quality reads coming off the instruments immediately, and made it actually very, very affordable to actually allow you to do all kinds of population genomics in greater numbers. So when we talk about the whole genome sequencing, right, in this case, in this context, we really wanted to actually sequence a lot of samples, and we want to actually ascertain as much as possible in terms of the genetic variants, right? So people have been, used to sequencing the whole human genomes with the short-read technology in the last, 10 or 15 years, right? These short-read-based whole genome sequencing are largely capturing the SNPs and indel information. So therefore, there is quite a bit of information that's missing, right? According to what we understand now, after we have the more complete, understanding of the human genome, which is largely enabled by the PacBio HiFi technology, right? So in our vernacular, we're actually, when we talk about human whole genome sequencing, we're really talking about not only the capturing of the SNP and indel, small indels, right? We actually allow you to capture a large swath of other genetic variant types. For example, structural variation, DNA methylation information, information related to the segmented duplications, right? Dark region genes, those areas which couldn't be captured by the NGS, and tandem repeats, and plus you can actually, very nicely phase your genome into different haplotypes. So when we talk about whole genome sequencing, we're really talking about right off the bat, you're actually generating high quality reference genome already, right? You can actually allele specifically resolve the different haplotypes, and you can actually essentially capture all the known variant types, not just capturing SNPs and indels, which you could typically do only with the NGS technology. So the whole genome sequencing concept is really a totally different concept, because we actually deliver a whole class of new class of whole genome sequencing using the HiFi technology. So the HiFi sequencing is really built for precision health initiatives in the context of population genomics, right? So in this case, the high value information brought by the HiFi-based whole genome sequencing can really enrich the pre-existing short-read data set. As you probably can appreciate, there's a lot of whole genome sequencing PopGen project started with the short-read-based technology, and you can understand or appreciate that the information gleaned by the short-read technology is quite limited, as we saw in the previous slide, right? So the information from the HiFi whole genome sequencing can really improve upon the short-read data set, your better understanding of the genotyping, the variant calling, the representation of genetic diversity, right? And also it has a tremendous long-term data utility values, because these genetic variants that you capture right off the bat with the HiFi technology is very comprehensive, right? That basically allow you to maximize your information, digging. You can actually mine the gold mines later on for years to come. And the cost effectiveness and the scalability of our workflow has been demonstrated through the project like All of Us in the United States, which is a gigantic, PopGen sequencing project in which they wanted to sequence thousands of, hundreds of thousands of samples using the long-read technology. And currently, at relatively shallow sequencing, right, depths, as we have demonstrated, you can actually go through almost 4,000 whole genome equivalent genome sequencing samples on the Review system per year. And this actually can be fueled by the secondary analysis from PacBio as well as Google. And obviously, you're capturing high quality data from high whole genome sequencing, as opposed to these other noisy, long-read technologies, which all claim can do the same thing, but not really true, right? And this has been repeatedly demonstrated in the literature, as we have witnessed. And hi-fi technology is really more scalable, less expensive, and easier to actually get data and handle data. And storage, data storage is also very, very economic, and this allows more streamlined analysis as well. So what does that mean in terms of cost effectiveness, right? From a populational, genomics project standpoint, and you can see essentially here I'm laying out two different scenarios. On the right-hand side, okay, you can think of a project start off with building very, very good quality reference genomes. So in this case, a typical 30x sequencing depth will get you a very, very high quality Pan-genome, right? This is something we're going to be actually witnessing from Dr. Xu's talk. And you can see that the performance in terms of capturing all those variant types, comparing to the ground truth, is extremely high quality. And on the other hand, right, if you do not actually already started with the PacBio sequencing, right, in this case, you want to actually sequencing a lot more samples, and you can think of experimental design in which you can actually bring in pretty good large number of samples using the PacBio shadow sequencing, in this case 10x sequencing coverage depths, right, which can somehow cost effectively complement the existing short rate data you have already garnered from the Illumina sequencing technology. This basically allow you to phase all your short read data, boost the structural variation detection, call variants that are actually in the challenging areas in the human genome, right? These are typically medically relevant genes. You can actually profile the tandem repeats, and you can actually capture methylation information, right? So with this kind of a shallow sequencing, which is going to save you a lot of money, right? You're actually getting pretty good performance compared to the ground truth. So, and we also allow you to do this kind of a thing in a high throughput fashion, meaning you can actually bring in automation systems as opposed to handle in a manual fashion, which oftentimes potentially introduce errors in your workflow, right? So currently, we actually allow you to process your DNA extraction from the sample, right, using our nanobind HD kit, and then followed by the automated sample or library prep on the Hamilton system. And once you get that done, you can actually put your library prep onto the system to be sequenced, and you can see you can easily process up to 4,000 samples in a single year at a coverage about a third of typically what you need with the whole genome sequencing if you want to get very good quality reference quality. And we also provide a slew of software modules to actually allow you to tap into all these different types of variants, which is missed by the short read based technology. And before I hand it off to Dr. Xu, I'd like to make a final announcement. We're actually offering a summer grant program, in which case you will get a chance to actually submit your proposal, scientific proposal, to tell us what you wanted to do in terms of using the long-read Review system, as well as the recently launched short-read technology called ONSO, which is based on SBB technology. So capture this information when you get a chance, and then submit your grant proposal to us. So with that, I would like to hand it off to Dr. Xu. Welcome.
Thank you. Can you see my slides now? Hello?
Yeah, we can see it.
Okay. Good afternoon, everyone. So first, my thanks to PacBio for the invitation, and especially from Dr. Chen and the the team, right? Many members, and it's my great pleasure to be here online to present probably just a brief introduction of our CPC program, and today's slides are mainly based on our recent publication in Nature that also represents our latest progresses in our research on this topic, Pangenome Reference, Chinese population. So my presentation also is on behalf of our CPC members. So firstly, I give a very brief introduction to the context and also some very brief history. So many of you are aware this year is a very special time. It's actually the 20th anniversary of the complete of human genome project. So that was a big milestone, and but this year also probably another one. But if we trace back to more than 100 years ago, actually that was the field of I myself was training. My training as a population genetics. At that time, we didn't have any genomic data, but our people in this field, I have to say, those great population geneticists that already developed quite a well-established theory to study the genetic diversity within the between populations. And since 1990s, people, actually 1980s, I would say, people were discussing to sequence our own genetic code, human genome. So that took about more than 10 years, and eventually it was completed at the very beginning of this century, and followed by the HyperMap project. That was actually targeted at the GWAS, genome-wide associative study, actually. So because at another time, the whole genome sequencing was so expensive, so it was infeasible to sequence everybody's genome at that time, but genotyping was possible. So since 2005, the first GWAS paper was published, and until now, that was 18 years. So we witnessed these progresses, right? And actually, our Asian scientists also initiated our own SNP project. We called it the Pan-Asia SNP project. It also took about 5 years, and I have another slide to share with you to show you very briefly also. We characterize the Asian genetic diversity based on the genotyping array. That was actually the first generation microarray provided by Affymetrix at that time. And in 2008, the International One Genome Consortium actually launched this project, KGP. So this project actually demonstrates the rare variants in our genomes, and also from another time, even the GWAS people started to be aware of the importance of the variants, rare variants in disease mapping. And the subsequent efforts, I would say, after these three internationally collaborative projects, HGP, HyperMap, and One Genome, and also include our Pan-Asia project. And the most recent efforts in human genomics over the last 10 years, most of those efforts are pretty regional, geographically regional. So, for example, like UK 10K and now UK 500K, and all of us, and Singapore 10K, and probably now 100K. In China, we also have our Han Chinese 100K project. So those are all nationally or regionally efforts. So this is actually a big shift or change of the research collaborations in human genomics, I have to say. So that was also because the changes of the international political circumstance, I have to say. But that was not good, but that's the situation. That's the reality we are facing to. Okay. Anyway, so we are facing to the paradigm shift in human genomics. So this year we talked to this much about this situation. Now it's close to the end of this year. So many people in human genomics or human genetics are aware of this kind of paradigm shift from single linear genome reference to pan-genome graph reference. But I would say the practice or the practical research of applying applications of the pan-genome graph reference in both evolutionary study or medical studies are still relatively rare in the field. The reason, of course, the first reason was our current or the latest reference genome of the humans, GRC38, missed quite a proportion of the sequences. Although in last year, 2022, there was telomere to telomere technology available, and also there was a real complete genome sequenced, right? CGM13. But so far, this kind of study are still remain in the reference construction. Applications in human genome studies regarding medical or evolutionary study, population genetic study are relatively rare. It's just mentioned again. But anyway, so there are two papers. There were two papers published in Nature. One was by the US and the UK and the European team, right? HPRC. So that was also well known now, and we also published the Chinese, the first, also the draft, right? Pan-genome graph reference draft based on Chinese genomes. So that's the currency of the population human genetics. So this slide I would like to show you why graph genome reference is valuable, I would say, or why we really need graph genome reference for even the data analysis, right? The reason is that any available sequence technology, right? So far, no matter whether it's short-read-based sequencing or long-read-based sequencing, we extract the DNA from one individual cell or individual blood samples. So we have no way to sequence a whole genome at one time, right? So we need to create some library, right? Libraries, and then we sequence those libraries to short arrays or long arrays, anyway, sequence arrays. And then we need to map those arrays back to our references. So that's the typical practice, right, in our data analysis. So suppose we have only a single linear genome reference. So we project our sequenced arrays back to the reference genome. So the probability is that we can map the majority of our arrays, right? Sequenced arrays to our references, so no problem. But there are still quite a lot of unmappable arrays remained. Even we spend money to sequence those materials, DNA materials, the data are there, but actually we didn't use that, right? Those unmappable arrays. So we have those mappable arrays and for downstream analysis. So that's the practice. But suppose we have a Pangenome graph reference, so the practice the similar. So we sequenced our libraries and we try to map back those sequenced arrays to our reference, but now we have a Pangenome graph references like this, right? A Pangenome graph reference means we have multiple linear and entangled and very complex graph represent the human genome diversities of the majority of the population samples, right? So the probability that we collect or capture those sequenced arrays is much higher than linear geographical linear genome references, right? So that's actually the rationale behind this shift, right? Pangenome, the genome shift, reference genome shift, right? From linear to graph genome, right? So that's actually very simple. The term of the Pangenome is not very new, right? It was proposed in 2005, the first time based on the literature review. In 2010, actually, Li Chang et al. And his colleagues actually also based on short array sequencing, right? Assembled several human genomes and found some lower sequences at individual level. So they did de novo assembly, right, based on short arrays sequencing data. So since then, most actually over the last 10 years, right, many efforts have been made to create or produce individual personal genome, right? Personal genome, high quality personal genome. First in Western populations, right? Samples, but in Asia we also have those available. AP1 is a Korean individual, NH1 is Han Chinese, HX1 also Han Chinese, and these two in red, ZF1 is a Tibetan de novo sequenced genome. That was done by our group and collaborated with Bin Shu's group and also the local collaborators from Tibetan University. And we also sequenced another minority genome TG1, it's a Tujia ethnic group. So, and actually recently there are also some other more personal genome in de novo sequencing or de novo assembly style published. And then those are still personal genome or linear genome, right? Then shifted to the graph genome. The Pan-genome idea is actually very simple, since we have diversity among different individuals and also between populations. So we sequenced every genome and we identify some variants, right? And besides the single nucleotide variants, we also have many structural variations. So those structural variations and also small version indels, including indels, right, are different if we compose them together. So we would like to observe actually different lengths in the whole genome size and also in the local regions. So in this case, if we just use one single genome or linear genome to represent the major composition of the human genome reference, so that would be a problem, right? So we collect many different individual genomes and we try to cover or to present the diversity in a graph style. So then we categorize the different genomic regions. Probably some regions, different individuals actually share the same, very similar pattern or identical pattern. We call that the core genome. But the other parts, probably the majority or quite a few or quite a proportion of the samples share this segment or these sequences. We call that probably some common segments or dispensable genome. And but there are also quite large parts of the genome. It appeared in just one single genome. We call that the very private sequences. So collectively, and we have those days different categories called the Pan-genome, and we try to use those very complicated or complex joint graph as a Pan-genome. Before the availability of the technology of the long-race data, actually people also made quite a lot of efforts to create a Pan-genome for human data based on short-race sequencing. For example, those are publications, right, based on African or global population, no, African populations. And this Dr. Wei Caochun also and his team and developed a pipeline to assemble Pan-genome based on also short race and also Han Chinese genomes. And Lori Bang from Hong Kong, they also built a Chinese Pangenome of about 500 individuals. And the reason, right? Again, we explain why the Pan-genome is necessary, because again, the single linear genome reference actually missed a lot of population diversity, and short race also have its own problem, right? Short race sequencing has its own problem because the assembly is not so precise, and phasing is another problem. Okay, so there are many technical problems, so that if people would like to apply the sequence race mapping, so this imperfect reference genome would lead to a lot of problems, right? So another reason, our argument is also our own argument. The genetic diversity really matters, right? About 10 years ago, we analyzed or compared, right, our Asian genome diversity and the one-cell genome data, we concluded that the one-cell genome data do not cover well the Asian diversity. Of course, not true, and so far is also still true. And because the Asian country and the Asian populations, the population size is pretty great, not very large, huge, right? And also we have very diverse ethnic groups living in different, very diverse geographical regions, and including those islands, right? Or that's very remote villages, mountains. Okay, so that's also created a lot of genetic diversity. And the Asian people's history was also very long, so that also preserved a lot of very ancient genetic diversity. But the situation, the reality is that although Asian has a lot of genetic diversity, we also have enormous population size, but relatively compared with, especially compared with the population genomics of European ancestry population samples, we have less than 10% of genomics in the entire data bank, say, and in the whole human genomic data database. And especially regarding the GWAS catalog, the majority of the discoveries are in population samples of European ancestry. So there must be some ascertainment bias in disease mapping. So this issue was also very well known even before my presentation. And I just mentioned we conducted a study or project, initiated the project, Pan-Asia SNP project since 2005, and we published this science paper in 2009. We actually collected about 75 population groups, which actually represent the very diverse ethnic groups in East Asia and Southeast Asia, and also South Asia, because we included India in our study. So as you can see, there are very colorful picture. Those colors actually represent the different ancestry, right? Different genetic ancestry. We analyze the genome-wide data. At that time, we have only 50,000 SNP genotypes available, but those are actually sufficient for our genetic diversity analysis, especially to understand population genetic relationship and their genetic ancestry composition. So as you can see, this is very colorful, and even each single population, for example, the Han Chinese, it's a mixture, right? And those populations in the Philippines, in the Thailand, in Indonesia, and India. So their populations actually are highly mixed, right? That also means a lot of hybridization or gene flow among different populations. And we also identified quite a lot of genetic clusters, right? So that's also, as you can see here, this maximum likelihood tree we construct based on genetic data, but we labeled their populations based on their languages people are speaking, right? So there is a very good relationship between languages and genetics. So that also means the cultural language or the other factors actually strongly influenced on the genetic composition or genetic makeups of different populations. But on the other hand, again, as you can see, even you look at each single population, Japan and Korean and Han Chinese, right? So you can see a mixture of different colors, right? Different colors represent different ancestry. So that's very complex. Also represent different differentiation of different populations. But taken together, you can say very diverse genetic makeups in Asia. Those all the data were still very old, but we have this basic understanding. So that's also the reason. So another rationale behind this understanding of the genetic diversity, or why we concern this diversity so much, that's because there's two examples, right? The previous study actually already identified the very important or key genetic variants for some rare diseases, right? Behind the rare disease, there are some very important rare genetic variants. For example, this CDA1, I see it was reported to be associated with anemia. But even for this so rare genetic variant, and some doctors or clinical trial can use this for diagnosis, but that can be useful or workable in some African populations probably. But if you apply this variant to non-African populations, it doesn't work because even in natural populations, those non-African populations have a high frequency of this disease allele, right? So that means a different genetic background can matters, right? Diversity matters. That's one example. This is very similar. PDF1A again associated with some pancreatic diseases and other diseases. Okay, that was done by a previous study. But if we look at the global populations, again, we observe this great differentiation between Western and Eastern populations. So that's why we said genetic diversity actually matters even in terms of medical studies or practice, right? So another example regarding, so this in the context of human local adaptation study. So this is also very well known, Tibetan high altitude adaptation. So we identified one deletion in their genomes, this 0.4 kilobase pair deletion taking place in Tibetan people in a very high frequency. Actually, more than 90% of Tibetans carry this deletion. And also, but the other population, non-Tibetan people, it's very rare or absent. You cannot observe it yet in the other populations. Even occasionally you observe such deletion, but you can trace back their origin probably from some gene flow of Tibetan people. Another example is this gene TMEM247. So this gene is the function is not so well known, and no much study have been done, but there is missense change or long synonyms change. So very likely this is a very important mutation or variant, right? So we also have some evidence to support those deletion or non-synonymous mutation associated with high altitude adaptation of Tibetans. Okay, so this example again, actually the message, right? The message behind is that this diversity or population differentiation is very essential in both medical studies and evolutionary studies. Okay, we can also apply this kind of population comparative analysis to any other populations. For example, the Han Chinese, the largest the ethnic group of the largest population size in the world, right? So we can even identify this particular gene FADS2 between northern and the southern Han Chinese. So generally, people treat Han Chinese as one homogeneous group, right? But they even just the southern and the northern population, they also show the very distinct differentiation in some particular important genes, right? So regarding the copy numerations, again, in Han Chinese, we also identify some functionally important or potentially important gene or copy numerations. Globally, in the entire Han Chinese population, probably the frequency is not high. For example, less than 0.5%, but in some very regional Han Chinese population, for example, in Fudan Han Chinese, this particular copy numberation can reach to a very common or high frequency, more than 10%, right? So this actually have great implications in our disease mapping or to do the clinical trial or diagnosis, right? So we actually deliberately evaluate the necessity to create a population-specific reference genome or reference panel. We use the Tujia genome as an example. The reason was that the Tujia is the ethnic group showing the closest relationship to Han Chinese among the 55 official ethnic groups, right? So the rationale was that suppose we find the necessity is high for Tujia specific reference genome, then of course this apply to the any other ethnic groups is also valid, right? Okay, so we evaluate this from 3 respects. Variant calling, especially this example, right? PRSS gene region is also have a very complex structural relations. Suppose you don't have this population specific reference genome, very likely you applied very routine NGS based mapping or short range mapping, right? So very likely you will have a wrong coding. So your variant calling or genotyping would be erroneous. A lot of errors occurred, right? So variant calling. This is we demonstrated this example, but we have actually definitely a lot of such examples, and we also evaluate this for evolutionary studies, medical applications. So of course, the conclusion is that the population-specific reference genome is necessary. Okay, so that's also the reason, right, why we initiated this Chinese Pangenome Consortium to apply long-range sequencing, because short-range sequencing is not so powerful for de novo assembly and structural relation discovery and genotype coding, right? Especially regarding the copy numerations. So we sequenced actually 68, but eventually we found there are 10 samples were not in high quality. So we just excluded them. So eventually we published this only 58 core samples of called that. So of course that means 116 haplotype phased assemblies. We released this eventually. But although the sample size was not large, but it also represent the very diverse ethnic groups in China. In total, we have 56 official ethnic groups in China, right? So now we cover the 36. Okay, so of course this is a collaborative efforts from many different universities and team members. So I will give you a very quick impression, right? Those numbers. Of course, those are just some numbers regarding the data quality. So the average continuity, measured by N50 of the race, right, is 35. The context, right, 35 megabase pair. And on average, we sequenced our genomes to more than 30x. And the coverage compared with the latest reference sequence, the GRCH38, more than 96%, and even compared with T2T CHM13 is more than 93%. So those two numbers are actually very important, right? So for each single sample, we can identify about 60 megabase pair sequences. Those are not being discovered in GRCH38. So that means, so this, if you have some idea of the chromosome size, right? So this is roughly equivalent to chromosome, the entire chromosome 21. So just imagine if those missing sequences, right? Those are missing sequences from the reference genome are all genes. So there are probably several thousand or 10,000 genes, right? So that's really remarkable. So even compared with the T2T CHM13, we can still identify 24 megabase pair missing sequences. Okay, so this is the basic idea, some impression, right? So in case there is no such efforts, or actually, and previously we missed how much sequences in our analysis. I will talk about this again in GWAS, in the context of GWAS or disease mapping. So this is a simple comparison of the data quality with the HPRC's efforts, right, the paper. So on average, so overall, we have slightly lower N50 genetic genome continuity, right? But because we have a limited financial budget, right? So we didn't sequence our genome to very high coverage. So that was the main reason. So that the reason was not because of the analysis. So again, numbers. So I just said about 70 megabase pair sequences missed, right? So in these missed sequences, about 6 million small variants can be identified, and about 34,000 structural variation can be identified. And very importantly, these missing variants or sequences actually encompassed about 1,367 protein coding genes. So why I emphasize this number? Because that means those missing sequences, they're probably also functionally important, right? So generally, protein coding genes also means some very important product potentially, right? The functional and also probably associated with some our human phenotypes or diseases. Okay, again, numbers. So of course, there are some hotspots or enriched regions with those missing sequences. Of course, as you can expect it, most of the missing sequences are in the centromere, telomere region, of course. That's I expected, right? So another question is that since there is already HPRC effort, right, internationally, so why we also must do a regional efforts for East Asian peoples? That's because even the HPRC or their future studies very likely miss some East Asian diversity, as I just demonstrated to you. Asian populations, Asian peoples actually harbored a lot of genetic diversity that cannot be covered by the other geographical regions, European or American peoples, right? So we identified some East Asian specific sequences. Of course, I must clarify that this comparison is what was done between HPRC and our CPC Pan-genome data. So this, of course, this so-called specific is relatively specific. That's just the comparison of the two data sites. But in this case, we did identify about 200 megabase pair sequences specifically to East Asian, and I just explained, right, the Pan-genome composition, core sequences about 5 MB, and the common sequences about 72 MB, and the singletons, or you can treat them as private sequences for each individual, right? Collectively about 62 megabase pair. So this is the distribution of those non-reference sequences. Sometimes we call them novel sequences, right? But this is again a relative term, right, or concept. And across different linguistic groups, again, we have about 9, right? 9 ethnic group, language linguistic groups. So as you can see, as we sequence more genomes and the increase of the singletons or private gene non-reference sequences is very clear, right? Apparent. Okay, again, 66 million small variants and 34,000 structural variations in our CPC data site. So we highlight some East Asian specific novel sequences in terms of the gene, right? The genes and gene annotation. We highlight this one, CYP2D6. This gene actually involves a lot of medicine metabolism, so it's very essential. But East Asian and non-East Asian people showed a pretty remarkable differentiation. So this also has great implications in future medicine practice, right? Medical practice. Okay, we also, because we haven't done any functional assay or functional experiment, so to try to understand the functional potentials of our non-reference sequences identified from CPC data. So we applied evolutionary studies analysis of our data. The rationale is that suppose there are some adaptive evolutionary signatures occurred or observed in our data. So it means potential functionality, right? So the reason that we can observe, right, those adaptive signatures because these sequences are functional, Otherwise, there is no any gene environmental interactions. And so that's the identifications or the observations of those adaptive signatures means potential functionality of those non-reference sequences. Okay, so the more important gains of the long-range sequencing or this efforts of de novo assembly of the whole genome using this Pan-genome approach was that we have opportunity to dissect the fine-scale genetic structure, the structural relations, right? So there are some examples I will quickly show you. So this PRSS I just mentioned, right, in our evaluation of the TJ1. So as you can see, this relatively clear structure we can observe from this Pangenome references. This is another example. It's also very outstanding region, geographical region, genomic region that previously already reported. It is associated with schizophrenia. But as you can see, this we call that the bandage graph, right? This just demonstrate their structure configuration of those this gene region, right? In different individuals, there's different individuals. This is the composition of the different components, the diverse components of this structure variations. So this is an example of the scape there. So there's also some very interesting findings in the in terms of human evolution. So I just mentioned at the very beginning that in the old style, right, or in the old paradigm of the based on short array sequencing, we had a lot of unmappable arrays. We didn't analyze that, even we sequenced them. But now we have this opportunity to recapture, right, those unmappable arrays back. And we actually anticipated those unmappable arrays probably something very interesting hide inside, right? Especially those sequences probably derived from those archaic hominids, right? For example, Neanderthal, Denisovan, or some unidentified species, right? So indeed, we identified quite a lot of such sequences. As you can see, this is actually considerable, right? This size, 600 megabase pair collectively. So we further annotated those sequences. We actually found them are related to some very important functionality. Characterization, UV response, DNA repair, immunological responses, even lifespan. Of course, we just search the database and try to collect the information that try to correlate, right, the sequences and their reported functionality. But anyway, so this gave us some encouragement that more studies should be done to discover those hidden stories in our genomes, right? Or for our human beings. Okay, we also developed some algorithm, the tools during this project and also before. And we also created some database, and especially for future study, I have some perspectives to share with you. Again, the rationale or the real reason or the very practical reason behind this efforts is actually there is a great challenging problem in human genomics, especially regarding the genetic basis of the complex disease. So this is a very common challenge in the whole field, missing heritability. Because we really know many complex diseases such as schizophrenia. Based on the family data, we can estimate their heritability is as high as 80%, more than 80%, even autism, right? Even height. We know the genetics contributes a lot, great, more than 80% to those complex diseases, but the last 18 years GWAS study identified a lot of genetic variants, but can explain only less than 20%. Most likely for a single study can explain only 5% or less of the genetic heritability. So that's the common challenges. So of course, people are proposing, right, some different models to explain this genotype and the phenotype relationship. Traditionally, we have monogenic model and the association study, polygenic model, and very recently omnigenic model. That means in our genome, almost all the genes, genetic variants are related to our diseases, right? And some people also argue environment is very important, and the gene and environment interactions are important. Of course, I would agree, but our argument is that since we missed a lot of sequences, as I just mentioned, right? 70 megabase per individual sequences. So since you missed so much genetic variant, so much genetic sequences in your studies, so no wonder you have this missing heritability, right? So this is reasonable. So what's really missed in our GWAS study is especially structurations. So I just mentioned 18 years of the GWAS since the first paper published in 2005, and nowadays we have a very rich data set in GWAS catalog, but the majority of the GWAS paper just analyze the SNP markers. So this is actually the simple principle of the GWAS study or disease mapping. So we have unknown disease allele in some genes, but we don't know the exact locations, right? We even don't know the genes. So we just analyze those markers in the genome-wide or the whole genome sequencing data. So the hope is that there is some linkage disequilibrium between those. We analyze the markers and the potential disease alleles. For example, here we have this R-square 0.6. Very likely, we observed some statistical signals in this marker, and then we know this disease allele must be some nearby regions, right? So that's the reason. So that's the rationale, the principle. Very simple. But as I just mentioned, the majority, almost more than 99% of the GWAS study just focus on the SNP markers. So missed this structural variation, but the structural variation composes the majority of genetic variations in our genome. For example, the average genomic variation between any two human individuals, we usually use this number, right? 0.1%. But if you take structural variation into account, these differences between any two individuals increase to at least 1.5%. So this is the change of the magnitude, right? So you can imagine, suppose we recapture those unanalyzed sequences, especially structuration, and take them into account in our disease mapping. So there is a great increase of the probability that we capture those missing heritability. So that's the reason. So that's why I said again and again, especially this year, in different occasions and different conferences and meetings. So I said post-genomics was too early if you propose this. and also so misleading. Because if I just borrow the quote from Churchill, Sir Churchill, all right, the genomics it is not the end nowadays, right? It's not the end. It's even not the beginning of the end. It is just perhaps the end of the beginning. Okay. So we also have some rough future plan. So this is not so precise, but this is just in our mind. And we just completed phase 1. So I just forgot to mention, besides the non-race sequencing, we also have this NGS sequencing samples. That's because the cost was too high for long-race sequencing. So for population studies, diversity studies, we still need not short-race sequencing for larger sample size. Okay, so phase 2, phase 3. So probably there is not necessarily this kind of the barriers, right? Probably we can do them together, and we of course we will increase our population size, and also T2T now is feasible, I would say. So at least we will do 56 T2T. Of course, you can guess this number because we have 56 ethnic groups. So at least one sample per ethnic group, right? Of course, again, NGS samples, and the more Hi-Fi and ONT ultra long data. Okay, so although I just criticized the proposal of the post-genomics, but indeed, since we identified some non-reference sequences or novel sequences, we do concern about their functionality. So, but now we have Human Phenome Institute established in Fudan University. So we have these facilities and equipment, right? Or approaches to do the high throughput phenotyping. So it's very likely we have this high throughput phenotyping technology applied to our CBC study to understand their functionalities of those non-reference sequences. Okay, so yeah, this is some numbers that just demonstrate to you how powerful of those facilities and the phenome studies. Okay, we also released this CDC Pangenome references for our community and for your potential applications. We also integrate the CPC and HPRC data and created so far probably the best Pangenome reference in the world for future or for current practice. Okay, of course, this study or this project or this entire consortium was the effort from many friends, many people from different universities. I appreciate them very much for their very helpful comments and also their efforts, right, contributed to this study. And especially I would like to thank Yekai, right, for his trust, and we actually did this project actually together, and this is very important, right? I'm a population geneticist, and Yekai has developed a very great methodologies. For example, he recently published as a Beijing this very wonderful tools for this wonderful tool for structuration analysis, especially for complex structuration analysis. Okay, so as I just mentioned, we're still doing some further next phase of the CPC. So please join us and let's do together. And I would like to stop here, and thank you very much for your attention. And you are also welcome to visit us at Fudan University. Thank you very much. Okay, I'm done.
Yeah, great. Thank you, Dr. Xu. That's a very informative talk. You can see that our audience is still sticking around, even we read all the time. But let's try to answer maybe a few questions. Due to the time constraints, I think we don't want to handle all of them. I think based on the overwhelming questions that flooded in, I think there are one very important concept I think would be helpful for you to address to the audience, and that is when you design this kind of a pan-genome project, right? This particular phase, how many samples you would recommend, right? In terms of sequencing through these, reference quality genomes to construct a very good quality pan-genome. So in that respect, obviously you need to take into consideration a number of things, right? For example, there might be some substructures in the community, right? The population size, you consider the admixture level. So taking all these into account, so what's your recommendation in terms of picking the distribution? The sample.
Yeah, I would say, this is a great question. It's actually a very important parameters in our study design. But I have to say there is no simple answer to this question, but we do have some consideration of our own source regarding this very, very, very important question. So I just mentioned, right, Dr. Chen, you just also heard my presentation. I mentioned for several times that the real gain of the long-range sequencing or pan-genome graph genome studies was those very rare genetic variants, right, or private variant for each single individual. So from this point of view, so we can calculate, we do some simple calculation. So how rare is rare? For example, less than 1%. Less than 1% means we need at least 50 samples because we are deployed organism, right? Human beings deployed. 50 samples means 100 chromosomes. So 100 chromosomes, we have this probability to observe a singleton in our data set. So that means 1%, right? So at least we need 50 samples for each homogeneous, I would say homogeneous population for this pan-genome studies or graph genome studies. So this is the minimum sample size. So from this respect, you can imagine our current draft genome is not sufficient to carry the our genetic diversity for the entire Chinese populations. Also, of course, for the entire global populations, and that's much less. So we need much more efforts to increase this population size. So this 50 samples is I believe it's a minimum. Yeah, it's minimum. Right, 50 samples.
Thank you for sharing that knowledge. Very informative. So I remember in your one of your slides, you also hinted at the value of the pan-genome in the context of medical, clinical aspect, right? So what's the value? So can you maybe make a little bit more deep comment on that? How do you see the pan-genome can actually contribute to maybe coming up with some kind of a clinical setting, clinical diagnostics later on?
Okay. So again, this is really a great question. So now is the time that we consider, we think very carefully about this question. So previously we have already done a lot of sequencing, first generation single sequencing and NGS for last 10 years since 2004. I don't know, 20 years. 20 years. And of course, there are a lot of medical practice, clinical practice using those technologies. But now, as I also mentioned in my presentation, people are aware of the importance of rare genetic variants. So among the rare genetic variants, the structural variation is, again, even more important. And in the future, medical practice or clinical practice, I believe long race-based structural variation identification, discovery, or clinical trial would be very valuable, especially in some very special diseases, for example, birth defects. Now we have a lot of literatures, a lot of evidence to support that the birth defect was caused mostly by structural variation. And those structural variation, many of them are just or de novo mutation or very rare in some very special families. So the precise identification, precise genotyping, precise detection is very important. I believe the non-race sequencing technology would play a very important role, essential role in this kind of applications. Yeah.
Okay, great. Yeah, thank you for that valuable insight. So thank you, everyone. I think we really have to end here because we're really running over time by quite a bit. And thank you, Dr. Xu.
I ran out of time, right?
Thank you for your very, very interesting talk. And so for those of questions who will not get answered, we will actually take care of them later on by email communication. And again, we'd like to thank everyone for your time, and that this recording will be made available later. And everyone have a wonderful rest of the day. Thank you.
Thank you for your attention, and thank you again for Dr. Jian for your for sharing this webinar. Okay.
Thank you.
So bye bye, everyone. Have a good day.